Well, I hope it's awesome, and sorry for making your lunch later. My name is Jay Whitebear. I will be doing structural characterization of natural antisense transcripts, nanopore sequencing, as you mentioned. The white bears on the slide are irrelevant to the talk. They are a play on my name, two genetically different white bears. It's a joke I tell every presentation, and it's pretty bad, but so the goals of this presentation are to detect features of secondary structure by measuring nanopore data. Specifically, we're looking at natural antisense transcripts. We're trying to develop comparable methods to shape map and shape CE for direct RNA sequencing and characterization, and utilize the advantage of long reads and direct sequencing to examine secondary structure in more varied conditions, such as in vivo, etc. We use these methods to develop an unsupervised method to detect and classify anomalies, which would be different structural characteristics, and to evaluate transcriptomes, in vivo structures, endogenous modifications, long-range interactions, and more. These are the goals, of course. So for those of you not familiar with nanopore or direct RNA sequencing, the experimental overview basically is that there's a biological nanopore, which is just a very small pore, and the RNA are fed into this pore, and they use a motor protein to actually insert the RNA into the pore. And then you get a number of bases in each pore, and that's about 5 to 10 nucleotide bases in the pore, and those are referenced as Kmers here. For general reference, nanopore uses a 5-Kmer, or 5-mer, I should say. So we design experiments using a large number of reads to create large data sets that would not only be experimentally relevant, but also be large enough for it allow us to apply machine learning algorithms downstream. So here we use roughly 20,000 reads as unmodified controls to statistically measure the background noise in each in vitro sample, and 5,000 modified reads. So for SEN and IK2, which are the natural antisense transcripts, you see that we have greater than or equal to 20,000, just so that we have a large enough data set to have kind of relevant statistics from the machine learning. For the structure probes, we have two requirements. One, that they're small enough size to fit through the nanopore, combined with the nucleotide base without clogging the nanopore. And that is actually a very big issue. You can't just use any shape map probe or any shape probe with nanopores. And we also need to create a large enough reaction or impedance to be detectable in the current. So as it goes through, I forgot to mention this, that there's a current being directed at the RNA, and this is in picoamperes. And we measure that impedance as a characteristic of the nucleotide. In our case, we're using ACIM examined by Stevenson et al., which proved a very useful structure probe, which we'll discuss more later. So this is a sample of the data generated by Nanopore and processed using nanopolish, which is using a hidden Markov model to align the signals with each nucleotide, or Kmer, I should say in this case. And we have the key features indicated as above. Two of the really important features would be the event level mean, which is the actual signal going through the nanopore, and the dwell time, which is how long that nucleotide is in the nanopore. So after sequencing the reads, or sequence data is aligned with a reference sequence, which is just the sequence that you're using in your library. In some instances, nucleotides may not be read or the data is not aligned. These are indicated in pink and the negative ones. You'll see this most commonly in the 3' and 5' ends of any direct RNA sequencing for about 5 to 10 nucleotides, as the signal is kind of attuned to the RNA strand. And this varies over the number of RNA you're sequencing, the length of the RNA, and so on and so forth. So this is the overall nanopore workflow. After sequencing both unmodified and modified data and aligning the sequences, we can begin the analysis. So here we introduce Dashing Turtle, which is like the code name for our software, which is currently in developing to actually perform this entire pipeline. Now, there are several software models available, but none of them is complete. Some of them perform segments of the workflow, but what we're aiming to do is create something that performs the entire workflow to make it very accessible. So for analysis, we're looking at developing unsupervised outlier detection algorithms, focusing on both the base calling error and the signal analysis features generated by Nanopore. So the base calling error is something that's generated by the recurrent neural network that's proprietary to Nanopore. So it's called GLBIO, and GLBIO generates these errors that it detects as it tries to read which base is actually in the Nanopore. The problem with just using the base calling metric is that it fluctuates depending on the model they use. So it's a proprietary model. If they decide to change and use a different model, then you're going to get a different base call error on that sample. Additionally, because this is kind of a new experimental method developing these shape techniques on nanopore, you don't actually have the parameters established for the structure probes and the concentration of the structure probes that you're using. So there's no optimized model for this. And so this is something that we've developed in our experimental analysis along with our pipeline. Oh, I should mention the one the structure probe that we're using is actually acetylimidazole, and we're using it at 150 millimolar concentration. And this was determined after multiple testing. And then in previous work, these concentrations and this structure probe were shown to be very accurate with nanopore. You'll see with standard shape probes like 1M7 and 1M6, you get very strange or very diverse anomalous signals. And this is in kind of the unmodified versus 1M7, 1M6. So you'll see that the errors vary differently, but they're on the same sample. So what structure probe you use and the concentration of that structure probe are actually quite important. So to better understand these changes, what we really have to do is look at the kinds of base call errors that we're seeing in correlation with the concentrations of the structure probe that we are using. When we measured these, obviously acetylimidazole created the most significant structural changes. I have to also say that at lower concentrations, it wasn't quite as significant, and it was hard to detect it from unmodified. So you have a significant amount of background noise that you need to account for when you're using these probes. So again, with acetylimidazole 150 millimolar, we observed a near doubling of error rates, which was very nice because then we know that our structure probe is acting and that it is showing us something significant in the changes in the nucleotide. So from this, we developed the base call error profile, and this is pretty standard with some of the other methods in that we take the sum of kind of the insertion, deletions, and mismatches or mutations in the nucleotide sequence and divide that by the number of reads at that particular position of RNA. So each position, you're going to have a different number of reads just because you have a large sample and because the base call errors fluctuate. So in order to control for this, we take this sort of average. We then compared the base call outliers to SHAPE-CE, an experimental method that uses immunofluorescence to detect nucleotides modified by ACAM. So we're using the same structure probe in both experiments, and we detect outliers observed in a highly varied correlation with low accuracy. So one of the biggest problems that I saw was that people were trying to use base call to predict things with nanopore, and it works very well if you're using synthetic RNA and if you know exactly where your modifications are and on very easy data sets or kind of toy data sets. But when you get into like in vivo situations or in situations where you don't know what the structures are, it becomes a lot more challenging and complicated. And the accuracy was only about 20 or 30%. Oh, I should also mention that the underlying distributions are not normal and they display non-parametric behavior. So that means that you can't just say something is an outlier based on it being a normal function because these functions are not normal. And not only are they not normal, they vary by sequence. So in order to evaluate this a little bit better, what we did was look at the cumulative distribution function. So I subtracted the background distribution from the modified distribution, and we calculated sort of a delta F function of that, which kind of gives us a nice peak that you can sort of see a little bit on this graph slide. What we then measured these peaks. And then we applied kernel density estimator to the delta F, which is the peak distribution. And then we obtained a reasonable estimate of the probability of a modification at a given position. We calculated the accuracy at a given position based on localized peak detection with a 5 to 10 base range or Kamer size. And then we also looked at, the reason we looked at this is because previous work has shown that there is a constriction in the nanopore between the 5 and 10 nucleotides within the nanopore. And we obtained accuracies between 50 and 70% using this method in comparison to directly using base call errors in our prediction sets. So we then took the relative probabilities that we generated and used them in correlation with the signal analysis, which we discuss now. So again, I'll go back to this HMM model that generates kind of the signal and the dwell time that we were looking at before. And we want to analyze this data in such a way that we can observe changes in the current mean and dwell time produced by the addition of the structure probe. We assume again that large differences in these measurements between unmodified and modified sequences indicates a structural change. So we applied an unsupervised SVM trained on the unmodified data to predict outliers in the ACM data and generated associated class probabilities. And so this is demonstrated here. And then if we look at the TS&E of this, we see a much more uniform outlier detection across these different sequences. So we have E. coli here, we have SEN, we have HCV, IRS, tetrahymena, and the IK2. And for the unmodified, it's much more uniform, and we don't see quite as much diversion in the anomaly. So we measured the correlation between the base call error profile and the unsupervised outliers distribution using Mann-Whitney. And while we do have a correlation, it only informs the distribution and does not tell us much about the types of errors or how those errors contribute to determining the modifications. So we wanted to get a little bit more specific than just base call error. Instead of directly using SVM or other ML methods, outlier detection, to generate a reactivity profile, we take the probability of modification or class membership generated by the SVM using Platt scaling or other similar methods if you're using a one-class SVM, and use it as a component in a decision function. And so finally, we wish to pair our generated probabilities to a broader understanding of the structure and accessibility of the RNA. So I call this deconvoluting the structure and probe distributions with Dashing Turtle. So we created a supervised method using a 3-layer feed-forward network with a category embedding of k-mers using the signal analysis features and categorical cross-entropy to generate probabilities for class membership just in our supervised data sets or for our controls. And we examined performance on multi-class data for predicting only nucleotides at roughly 96.7%. So we have very nice kind of correlation between the signal generated and the nucleotides that we can measure this relatively, pretty accurately. We extended the multi-class method for a multi-output by adding notation for the underlying structure base paired or single-stranded using the signal analysis features. So we also looked at, is it base paired or is it single-stranded? Because something else we wanted to look at was, can we detect more than just, is there a modification, but the underlying structure? Is there some correlation between the signal and dwell time and the underlying structure of the RNA? Here we observed accuracy at roughly 97.5%. However, as you see in the graph here, the C's were overpredicted and may have been overrepresented in the training set. I think they were a little bit skewed here, or there could be some correlation between that and the non-Watson crick pairs. We're not sure, so we're still looking at that data. Finally, we combine all of these generated probabilities into a decision function that is able to predict nucleotide, single or base pair, and whether precision has been modified for better understanding of both the structure and accessibility of novel RNA sequences. So here we're just mapping our decision function where we take all the probabilities that we generated in the previous step for base calling and for signal analysis, and then we combine this with our trained method and we produce probabilities there, and we map it into a decision function which is currently in progress. So we're in the last phase of mapping the decision function for the maximum accuracy. And finally, we want to generate structure landscapes. So for natural antisense transcripts, it's assumed that they've form fully complementary bindings, but this hasn't actually been confirmed. So there's just an assumption that this is true, but we don't really know. So what we want to do is identify the structural landscape between monomers, kissing complex, duplex intermediate, or the full duplex assumption. And so what we can do is look at that individually for each nucleotide and see whether we have a structural modification and measure the accessibility of that position. So if we look at K-means and the Gaussian means cluster, we can see that when we add the structure probe, we get a separation of clusters at that, and we introduce additional clusters that aren't seen in the unmodified data. And this is for highly modified positions in SEN. So here, this is SEN 3' UTR at position 15. And then finally, what we like to do is determination of a dominant structural interactions, mapping individual reads and sample to observe the frequency of modification in a given position in a given sample, and generate a heat map to observe areas of high activity and therefore indicative of high accessibility or more open configuration at a given position. We want to measure also the likelihood of the overall reaction based on the number of reads modified in the sample, and that's some data that we already have with NANOPORE. Finally, I'd like to thank Miguel, IRCM, GLBIO, of course, and FRQS, as well as my PI and our collaborators.